To start, so welcome to the day 4 of the workshop. I hope you have already enjoyed the first 3 days. We will be switching gears today from full atomic simulations to coarse-grained methods, where our goal is to have an understanding of the most cooperative collective events, as opposed to the detailed atomistic events. Things that are usually beyond the scope of MD simulations, much larger systems and slower processes. So as you know, this will be the next 2 days. We start today. I will give a few definitions, fundamental concepts, in contrast to MD simulations where everything is like F equals MA and the rest is some visualization and number crunching. In the case of these methodologies, we solve things analytically, which means we have a unique solution for each structure, a unique solution which is the most probable types of motions that each structure undergoes. So I need to go over the theory to show how we get this unique solution. And we have a package, software package, which enables users to either use it or even modify it for doing this type of calculations. So today, we will essentially have in the morning 2 sessions. I will talk about elastic network models, collective or normal modes, definitions, assumptions. And then I will go on with comparison with ensembles of experimental structures and show some applications with ProD, the software that we would like to introduce in this workshop. And then after the coffee break, Tim and Ahmet will continue with the ProD overview and applications. So these are, for each of those different components, I can give you a few general references. By the way, these lectures should be available on the website, so you can see the references there. And for those of you who would like to go into more details, but I will try to do my best to summarize them. So the first, most important concept we would like to emphasize is that each structure has a unique dynamics. We call it structure-encoded dynamics. And the structure-encoded dynamics can be viewed at multiple scales, from quantum chemical molecular mechanics all the way to large-scale machinery, supramolecular machinery. So what I would like to essentially talk about today is what happens at those large scales when we have several subunits, several domains, monomers coming together to form some functional machines usually. So we are going to, for example, if we concentrate on this particular AMPA receptor, which is formed by many, many different components, what we are doing is to take the structure, we use the existing structure, and try to determine what are the most accessible movements. And the premise under everything we do is, of course, our goal is to understand the function, the mechanism of function. And function depends on structure. But the bridge between structure and function is dynamics. Proteins or biomolecules, they function because of their dynamic abilities. Okay? So suppose we focus on the AMPA receptor, the type of calculations the type of movements that we obtain are illustrated by this case. So this is a big entity. This is the transmembrane domain. And here's an N-terminal domain and ligand binding domain. And it's conformed by 4 chains, a tetrameric structure. And you can see that what we are showing here is a collective motion that is most probable given this, the input is this structure, and we are able to generate this type of collective movements. And of course, in the last 15 years, there have been lots of comparison with experiments showing us that what we're predicting are relevant to experimental observations. And there are many ways of probing what we're doing. For example, in this case, if you see that particular pairs of residues and the distance, you can see how the distance changes from, let's say, 7 angstrom to 50 angstrom. It's quite large. So in the equilibrium structure, they are, let's say, 30 angstrom apart. Now, if the type of experiments that can probe is you can have some sort of interaction between them. If you substitute cysteines, you can form a disulfide bridge. You can have so it is possible to probe them, and indeed that's what we've been doing to verify, one way of verifying. So essentially, what we are observing are concerted movements of signaling molecules. So many proteins are molecular machines, and here that's an example of a big viral capsid. Again, knowing the coordinates of the viral capsid as well as the RNA, the genetic material inside, we are able to predict what are the most collective movements that the capsid is going to undergo. Or we can do the same thing at the molecular level. So that is, for example, a subunit of a molecular chaperone, GROYAL, where you can see it's an ATP-regulated machine. And you can see here how the structure may change between open and closed forms. So these are all predictions starting from a single known structure. So to do all these things, we represent the structure as a network. That is the basic assumption to begin with. Why do we use network models? Well, network models allow us to model very large systems, collective motions, and long-time processes beyond the capabilities of full atomic simulations. And of course, when you use network models, the network model that we adopt reflects the major structural features. And for example, all interresidue contacts, the contact topology. And of course, we take advantage of many existing theories, well-established theories in polymer physics, spectral graph methods, etc. So essentially, what we do, if we go back to this structure, we map this structure, the viral capsid, for example, into a network model. And we examine what this network model is supposed to, what is being predicted. What are the relaxational mechanisms or collective equilibrium motions accessible to that network model? And as I mentioned, these are, the theory is, this is my now acknowledgement to Paul Fleury, a Nobel laureate from year 1974, who established many methods of polymer physics, and in particular the theory of rubber elasticity, which we have been actually adapting to proteins with some minor modifications. So everything we're doing is actually rooted in the original statistical thermodynamics theories of Paul Fleury. So here, now I start to go a little bit into more details. Suppose we have a given structure. We map the structure into this network model, okay? That's what we're doing. And the network, so I have a small movie here, which will describe what we're doing. So as you can see here, an illustration for adenylate kinase. This is the structure. We focus on the backbone. We focus in particular on the alpha carbons, okay? And all the alpha carbons within a certain interaction range, we connect. We connect by elastic springs. So here's our network structure. Having obtained the network structure, we do our analysis, like ProD does, to calculate what are the most accessible collective motions. We call them mode 1, mode 2, mode 3. There is a hierarchy of movements, and there is a superposition of all those modes. But the top ranking 1, 2, 3 already define a large portion of the observed dynamics. These are the slowest events. These are those surviving, dominating the overall dynamics. So that is, and again, as I want to emphasize more that a major utility is when you apply to large systems, very large systems, hundreds of molecules, where you can start to observe the collective events, how different subunits move with respect to each other. Okay, so you have several references again here. The original elastic network model that we introduced in 1997 was called a Gaussian network model for proteins. So here is a few, I would like to present a few definitions which will be useful in your software applications. So here's our model. Suppose this is our model. And we have a bunch of nodes. Each node represents a residue. You can change that, but in the default is that each node represents a residue. And it is located at its alpha carbon. So we have position vectors for each node in a given three-dimensional space. Or you can think of the position vectors here are I, R, J, the equilibrium positions. And you have instantaneous deviations from those equilibrium positions. Or this is the, let's say, equilibrium distance in your X-ray structure. And you have a departure from the equilibrium interresidue distance to Rij. So this is the change in interresidue distance. So whenever you see these deltas, we call them fluctuations. Maybe this is not the best terminology, but it's like the departure from the equilibrium interresidue distance, which you can express as a difference between the departures from the equilibrium positions, okay, of the individual residues. Very simple. So what we are always trying to predict is what we have a given crystal structure. We have a given NMR structure, equilibrium structure. And we are trying to understand what are the fluctuations from those positions that are most likely to take place. And more importantly, we are trying to understand what are the collective fluctuations, not only individual residues, but we do things collectively. So we use what is called this fluctuation vector. So for a protein of n residues, for a molecule of n residues, this is an n-dimensional vector. Or you can think of a 3n-dimensional if you think of the XYZ components. So we are analyzing the behavior of the fluctuation vector, essentially. Now, the simplest network model, actually what is in the polymer physics theory, was the Rouse model. That was what inspired us. The Rouse model is, and you can even say, oh, this is not even a network, because it is just a string of nodes and springs, as you can see. Okay? In this Rouse model, also called a classical beta and spring model, we have, again, you can think of changes in the distances between pairs of nodes. And you can express the total potential of the system very simply by a sum of harmonic potentials. Okay? This is very simple. If you have any questions, please do interrupt me. So that is, we just sum up the energy, harmonic quadratic energy, quadratic functions to evaluate the instantaneous potential using a certain force constant gamma here. Or we can rewrite, knowing our expression, we know that delta R12 is the difference delta R2 minus delta R1. So we can replace these things by these differences instead and multiply by the force constant. Now, the force constant is a topic in which Tim will elaborate further. But in the simplest approach, you can just take it as a uniform force constant for all residues. Okay? And then the most important ingredient of the theory is what we call what is called the Kirchhoff matrix, taken from a theory of actually electrical engineering for network circuits. And in this case, what this Kirchhoff matrix defines essentially is the topology of your network. So if you have n residues, you're going to have here a -1 if a given pair of nodes are connected, or 0 otherwise. And the diagonal terms are found from the negative sum of the off-diagonal. So why do we use such a Kirchhoff matrix? Where the thing is, if we take this one, and we post-multiply and pre-multiply by the fluctuation vector, and also by our force constant, well, this product, if you calculate this product, what you obtain is nothing else than this potential energy. So this, in other disciplines, they call it connectivity matrix, adjacency matrix. But the adjacency matrix, pre and post multiplied by the fluctuation vector, gives you the potential of the system, okay? And in a concise form, you can write it like the total energy is the force constant divided by 2. Fluctuation vector transpose, gamma is the Kirchhoff matrix, and the delta R is again the fluctuation matrix. And that was more or less what was known from the Rouse model in polymer physics. What we did is, okay, we're going to generalize this matrix to not only consider things that are connected along the chain, but also any tertiary contacts. Because as opposed to usual polymers, proteins are very compact, and they make lots of tertiary contacts. So these off-diagonal terms are not necessarily 0, but now we're going to substitute there -1 again if those pairs of residues are within a certain interaction distance, or 0 otherwise. And the diagonal terms are again found from the sum. What the diagonal term essentially, what does it represent? Now, if you think of this definition, can you think of? It's very similar to like Ramachandran or contact maps in general, the structure. But I was just mentioning here we have -1s everywhere where there is a contact. And the diagonal term is found from the negative sum of those terms in a given row or column because it's symmetric. So what does the diagonal term represent then? Exactly, the degree of the node. So in graph theory, you would call it degree. Physicists may think of it as a packing density, how dense is a given node, neighborhood, how many neighbors you have. So, and intuitively, you may also start to think that we are going to invert this matrix. The inverse of it, the diagonal terms, will give us a measure of the size of mean square fluctuations. Okay, they will give us. Now, if something is very high here, a number, suppose we have a 10 here, which means a residue is interacting simultaneously with 10 neighbors. When we take the inverse, this number, roughly speaking, of course, if you think of a series, it's going to be small. That residue that's very tightly packed is going to move very little, okay? So that is the idea. Of course, similar to a contact map, you have, you can already recognize, oh, this is most likely to be an anti-parallel beta pair of beta strands, or this could be parallel. I think I need a cup. So that is, let me. Now, the most important, the only complicated equation here is this statistical mechanical average. Thank you so much. So this statistical, those of you who are familiar with statistical thermodynamic standard averaging know that if you would like to calculate any statistical mechanical average, you just multiply by the function by the Boltzmann factor, integrate over the conformational space, and divide by the partition function. Standard definition. So, suppose you substitute the potential that we obtain in this expression. So it's a multivariate Gaussian integral. And you don't, what you need to know is that the result is as simple as that. So what does this quantity represent? Delta Ri dot delta Rj is simply the cross correlation between fluctuations of residues I and j. Okay? So the cross correlations between fluctuations of residue I and j are simply proportional to the ijth element of the inverse of the Kirchhoff matrix. Okay, and this is just a constant, if you like, which is temperature dependent. Now, of course, we can write the same expression for the mean square fluctuations if I equals to j. So we have the diagonal element. Okay. Is everyone with me so far? Simple? Okay. So this is our key. The Kirchhoff matrix provides a complete description, two basic equations, and you will see how we are going to calculate actually the mean square fluctuations as well as the cross correlations using the diagonal and off-diagonal elements of inverse of Kirchhoff matrix. So this is just an application. For example, for hemoglobin, it's showing here that we, in experimental X-ray structures in the PDB, in addition to XYZ coordinates, you have also a column that describes the B factors, which is the B factor is a measure of the departure from mean positions, mean square fluctuation in the position. So this is the equation for the B factors. So now this gives us lots of, we can compare what we are predicting with the B factors. And here is an example where we did such a comparison. And many other groups have already observed that the level of comparison between what is being predicted, the mean square, and what is being reported is even better than MD simulations in many cases.
Is that Kirchhoff matrix there, or is that something different?
This one?
This plot here.
Well, this is not the Kirchhoff matrix. This is the cross correlation.
Okay.
So let me actually remember the Kirchhoff matrix gives you a measure of connectivity, who is connecting to whom, right? And this matrix is actually its inverse, because we said that the inverse of the Kirchhoff matrix here gives you the covariance matrix. It's called the covariance matrix, actually. So that one is nothing else than the covariance matrix. So this is the mean square fluctuation, this is the covariance. And that gives us information on who is correlated with whom, okay? So for example, for hemoglobin, in the literature, it's known that although it's a tetramer, they call it a dimer of a dimer, okay? Two of the monomers are very coupled. So we can see already two dimers very coupled and here. And they are less coupled with respect to the other two. So here, this map, every single point here gives us information on delta Ri dot delta Rj, which are the elements of the covariance matrix, how the fluctuations co-vary. Okay? Yes?
In such case, how is the heme group represented?
How is what?
The heme group?
Oh, in that case, you know what? That's a very good question. We can represent the heme group by a simplified model as well, the same resolution. So if we take one node per residue, let's say one node per 8, 10 heavy atoms, we can also represent this by a few nodes. That's one way to do it. What happens in very big systems, now we are talking, let's say we're talking about hundreds of nodes. And suppose you have one extra node. It's not going to change the behavior of the entire network usually. And that's the beautiful part of it. If you even repeat everything with, instead of uniform force constants, if you take full-fledged force fields and take the second derivatives to substitute the force constants, you're going to have practically the same results in terms of the slowest the most collective ones, because they are not defined by the details of the structure, but the entire architecture. Okay. Talking about the B factors, just a caveat. If you think of a crystal structure, the way a crystal structure is actually resolved, you have your structure surrounded by many replica, right? And you may have some what's called the crystal contacts here and there. And your B factors may be biased by those crystal contacts. So in many places where we have observed a departure between our predictions and the reported B factors, like this was one of the cases, we have seen that there are actually those intermolecular contacts which are nothing else than things that happen in the crystal structure, not under physiological environments, of course. And these things, for example, these particular regions, would give rise to, although the theory predicts here quite a large mobility, when we plot the mean square fluctuation as a function of residue index and a large mobility, we saw that experimentally observed values are much lower. And then we repeated our calculations by taking now all the structure, the crystal environment within. And now we could see that for the central, those places that make contact, the theory almost very closely reproduced the experimental. So the theory became as wrong as the experiments, okay, by taking the environment. But that's a good verification of the realism of what we are predicting, actually. And this has been repeated by many, many groups, of course, applied to hundreds of proteins. Now, one last thing I would like to, a most important concept I would like to give. Well, we are predicting the mean square fluctuations, the covariances in residue positions. That's so far so good. But we would like to do more than that. We would like to understand what is called the intrinsic character of the biomolecule. What are the most cooperative, the most collective events, the most concerted events that are most likely to take place in this architecture? And this is done by a normal mode analysis. Some of you may be already familiar with normal mode analysis. It's just you decompose the observed movements into an ensemble of many, many modes, some of which are more important than others. The lowest frequency modes, they have a higher weight given by inverse eigenvalues, so we will get there. So suppose now you are considering how a given, the motion of a given residue correlates with another one, okay? This is extremely important in the case of signaling proteins, for example, allosteric events. So we want to understand how they are correlated. Now we're going to write it as a sum of many modes, okay? Contributing modes. So K is the index for different modes. Now remember, we already wrote this equation. And we said that this cross correlation is simply the ijth element of the inverse of the Kirchhoff matrix. Now what we are simply doing is the eigenvalue decomposing that matrix or its inverse and expressing it in terms of its eigenvalues and eigenvectors. Okay, so replacing this by its eigenvalues and eigenvectors. And we have a summation over eigenvalues and eigenvectors here. Each individual term here represents the contribution of mode K, right? Now, this term in brackets, it's a matrix. Because you have a vector and vector transpose, it's a matrix. So if you sum over all the modes K, the summation is simply that gamma minus 1 matrix. So essentially, we can calculate from here what the contributions of individual modes are going to be, what the contribution of individual modes are going to be to the observed covariance or to the observed mean square fluctuations. So that is the idea. And this is extremely important. Why? The movements in the slowest modes that we observe are actually very relevant to function, and it has been observed in many applications. So we would like to dissect, the dynamics to extract. It's like a principal component analysis, essential dynamics analysis of your MD trajectory in same spirit. But this time we don't need a whole trajectory. We just use the connectivity matrix, okay? And this is just to give you examples. For example, if you have the this is the application to HIV protease. If we plot the softest modes, and what we are plotting essentially, let me see if I have it here. What we are plotting essentially is this eigenvector multiplied by eigenvector transpose itself and the diagonal elements of it. Remember, the diagonal elements were the mean square fluctuations. If we plot this one, we have a distribution of motions for all residues in the slowest mode. And we can already distinguish what is the active site, what is the recognition loop. So peaks represent what? the residues that move the most, right? So these are the recognition loops. You can expect them to move a lot and then accommodate substrate binding or ligand binding. And the active site usually, or a catalytic site, enzymatic site, we observe them to be at minima usually, because of they need to be very precisely positioned and everything revolves around them. Or the hinge sites. These are, for example, this is my hinge site. The hinge site doesn't move in space, okay? Everything around it moves. So we obtain information on such mechanically important sites. And also we can look at the fastest modes and the other extreme end, where the peaks will refer to regions that are centers of high energy localization. So these are really very, these are points you don't want to interfere with. And these usually serve as the folding nuclei because they have accomplished such a tight packing that could not be accomplished at early stages of folding, unless it took place at early stages of folding. So essentially what we are doing is the following in terms of a more physical description. We start with the equilibrium structure, and we are trying to extract the softest movement, the slowest mode, or the easiest collective motion, which is along that direction, for example, in this case. So we want to, suppose you perturb your protein, you bind a substrate. What is the natural way your protein is going to respond? Which degree of freedom it's going to exploit to accommodate the binding? So it's going to be most likely along its softest intrinsic motion. Are you with me? Yes?
What do you mean by softest?
Softest is, if you think of an energy potential, it may be very steep or it may be very smooth like that. So soft modes are very, for a given deformation, the energy cost is small.
But I guess what I'm wondering is where is the energy function being derived from? Is it from some theoretical considerations, or is it from the?
So let me see if I have the in the next slide. So after 2 slides, I will show you, okay? So this is, and if I don't answer your question, you ask again.
Can I ask another quick question?
Sure.
Is what we're doing here right now basically taking linear combinations of the
Absolutely, absolutely.
to derive the modes? So the eigenvectors
No, linear
themselves don't correspond to modes.
The eigenvectors are the modes themselves.
They are the modes. Okay.
Yes, eigenvectors are the modes themselves. Each eigenvector is associated with an eigenvalue, and the eigenvalue is a measure of the frequency of that mode. So if the eigenvalue is small, it means the frequency of that, the curvature of the energy surface in the space of that mode is small. So it is a soft mode. It's an easy motion. Is it clear? I'll tell more. So this is a summary of everything I explained so far. The Gaussian network model, we construct the Kirchhoff matrix, we evaluate cross correlations or covariances, and decompose them to evaluate the events in the individual modes. So far, this was all the description was about a graph, an n-dimensional graph, if you like. But if we would like to extend to 3 n-dimensional space, we use what's called the anisotropic network model, ANM. The idea is the same. We have the network representation of the protein. Now, instead of our Kirchhoff matrix, we have a Hessian now, where we have, again, eigenvalues and eigenvectors. But this time, everything is 3 n-dimensional instead of n-dimensional. And this is just one element, ijth super element of the 3 n-dimensional Hessian. I'm not going to go into details. But ideally, for example, the ANM mode 1, this is for p38 kinase. ANM mode 1 will be a motion indicated here by the arrows. And with Prodi, we're going to generate this type of Oh, if it was attached to my, sorry. So this is a problem now. Let's see. Okay, does it work? Okay, good. So for example, what ProD is going to do is, if we look at the ANM mode 1, it shows you what the direction of motion is. Of course, not only what the direction based on the arrows, but also we are going to see an animation of the movement. So in this case, it's like the separation of the two N-terminal and C-terminal lobes in mode 1. Or in mode 2, what's happening is some kind of a twisting motion of the two domains, two lobes with respect to each other. So these are like the most collective two modes of motions. If we go down the hierarchy to a high frequency mode, let's say mode 20, as you can see here, the red portions here are the most mobile, the portions that move a lot, right? So we can see only very few localized motions. Okay, it's not as collective. And the time scale, if this mode would be at least 1 or 2 orders of magnitude faster usually than the slow modes. And for each mode, maybe that will also clarify your question, we can think of each mode as obeying its own harmonic potential with its force constant given by the eigenvalues, okay? So this is the shape. So obviously you can see that that one is softer, that one is steeper, as you can see. Now the most important application is to when we want to examine allosteric changes in conformations. That has been a major utility. And we have seen that we have been comparing with experimental data, and it's so in particular we think for allosteric dynamics for signal transduction, this is one of the most powerful methods. So in the energy landscape where the molecule is originally placed, we identify what the first mode and second mode are. Okay, usually. Okay. Now, for so far, I have been just giving a general description. I would like to show a few applications here using ProD. And we are going to have essentially 4 sessions. The first one is about plotting the mean square fluctuation. And you will see how actually things are easy. So for the so we have a window for all ProD commands. And the first thing you do, and I'm going to follow exactly whatever is written on the slides to make things so that you can reproduce them later on your own. Okay? So we have, the first thing is to import ProD. What's that? All right. That's why, you know, I wrote everything here, because I know that if I do this in here when I'm on stage, I'll make mistakes like that. So the first thing you, of course, now this is a very easy command, this one. If you type ANM, ANM is the object that you define. Just to do the ANM calculations, okay? You calculate ANM. And we select here a protein, 1-COT, from the PDB. This was just randomly selected. This is a cytochrome C. And so it's going to retrieve from the PDB whichever protein you select. And you also specify, say that this is C-alpha atoms, okay? And you have now your object created. This is a protein of 121 nodes, 121 residues, and it already generated 357 modes. Actually, the number of modes is 3 times the number of nodes minus 6. 6 are the 6 external degrees of freedom, 3 rigid body rotation, 3 translation. Okay? And you have also the other. What is that? Why isn't it doing so? Oh, we haven't defined that. So that is, we need to define what the name of our object is. And to define it, we just write the name after A and M. Do you follow me? Now we have defined COT, our object, the protein we are going to work with. Now suppose we would like to view our protein the way only the alpha carbon of the protein. And I'm going to just pull that aside. So we would like to show the protein. So this is a very simple representation of our protein, in 3 dimensions, if you would like to see how it looks like. Or now, we talked about the square fluctuations, remember? So suppose we want to see the square fluctuations in another plot. And this is the command for showing, for viewing the square, all of them, the mean square fluctuations given by. So here you have the square fluctuations. So instantly we have already generated what the mean square fluctuations look like. And it's possible to compare with the B factors, for example, if they are available, as a function of raised to index. Now, as I mentioned, you can have the contribution of several different modes. So suppose you want to view here. Put that aside. Suppose you want to view the contribution of one given mode, the first mode, let's say, to the mean square fluctuations. In this case, it's quite small. You see in this diagram, you see the contribution of the first mode. Or if you would like to see the contribution of the first, let's say, up to 10 modes, that's what you obtain. So if you take a subset of 10 slowest modes, you can see that we already have those peaks that are essentially contributed by this subset of 10 slowest modes. And you can you can do the same. You can have a subset of, let's say, 100 modes to see where it falls. So that's how it grows. Gradually, the idea is all the modes, gradually they contribute to your mean square fluctuations and they add up. And it's possible to extract whichever you want. So you can label things. I will leave it there. But the easiest, simplest thing to do when you would like to examine a protein is perhaps to have an understanding of its ability, fluctuations, and what the contributions of individual modes are. Do you have any questions so far? Yes?
Can you visualize the strongest mode?
Yes, for example, or which one is the most important mode?
Of
the slowest?
Greatest contributor to the overall shape.
The greatest contributor will be always, by definition, the slowest mode. So here you can see by the green curve, although it looks small, it is 1 out of 300. How many did we have? Okay. So gradually, the contribution of the other modes gets smaller and smaller. So that is here. So that is what we have viewed so far. I'm going to close these things and move. You had a question? Somebody else had a question? Yes?
To what degree are we just looking
at a sort of a base representation of empty space in the protein when we're doing this kind of analysis?
I don't think
Are we looking at, are these motions or are these the possibility of motion?
these are the potential motions, but we are going to see the motions also. So far what I'm showing is this ability, if you like. This residue, I just removed it. That residue can move that much, etc. But then we are going to also view what are the accessible movements. So what the protein or any biomolecule, you can apply to DNA or any, what the molecule is doing is essentially recruiting these intrinsically accessible modes in response to perturbations to accomplish its function, etc. So we want to see what is its potential, what's its capacity, and we want to view them. Okay. So the next session, we are going to view these things in actually using VMD in an interface called normal mode wizard. So here I'm going to just, we're going to write our output into this new file named, I use the PDB code ANM NMD. Okay? So we write it there. So it's already there now. And we go to VMD. So I just want to, and we are going to here analyze normal mode wizard and load our NMD file. Let me see. So we already have now the structure. In this case, we are talking about, here you can see that in that NM box, we are now viewing mode 1, okay? And this is the molecule in mode 1. We have an idea of the motion it takes. You can view the animation actually in mode 1. What's the type of motion we are talking about? Okay. And as you can see, this is here superimposed on the, should I dim the lights a little bit further? Is there a means to do that? Is it better? Okay. So this gives you an idea of what the motion is about. And you can, for example, that's for mode 1. Suppose I select magenta color, and I would like to plot the mobility. So this is the curve for the mobility in mode 1. Which you can see. So if you would like to, the arrow colors are going to be the same as your curve colors. And you can have many different representations. You can select from here a certain residue. You can select a given residue. And it will be labeled here, as you can see. So as expected, this is one of the most, large motion, one of the residues that undergoes the largest motion. So this is the, and you can have many different options for the visualization. For example, let's see. You can represent, I'm going to hide the equilibrium, the original structure. So I'm hiding the original structure. This is the molecule that I have. And then I'm going to just represent it in terms of a network representation that I originally used. So you see this is the network representation of the molecule now. And you can change, for example, you can scale your sphere size. Let's make it bigger. It's going to be too big. But so that is another way of showing the motion. So this is the motion in mode 1, okay? And then suppose you want to see what the motion is in another mode, let's say mode 3. So this is another type of motion now that we are observing in mode 3. And you can select tube representation if you like, increase the radius of the tube. So this is a tube representation with the whichever width you like. So many alternative ways of viewing the motion. So essentially, to obtain this type of movements for a given protein is instant, But you can do that. I'm just illustrating the results for a small protein. Normally, you could do that for, of course, a much bigger system. As I mentioned, this is the major utility, and then you would see the domain movements. Okay. So essentially, this completes that session 2 as well. Anything you would like to ask before I proceed to cross correlations? Yes? The time scale. Well, this is always a problem, the absolute time scale. What we know only is the relative time scale. How, if we can adjust the time for a given mode, then we can calculate what the relative time scales are. But for essentially what this methodology is really doing accurately, I would say, is to give you an information on the mechanism of motion. Why do we want to learn about it? Because we want to identify, for example, hinge sites that we are going to target with drugs. We want to block this type of mobility. If it can't move there, it's not going to do its function. And usually many, many drugs are targeting the hinge sites, indeed. Now, what is the time scale of the motion? Well, I can't say anything about that. We don't have a quantitative measure. Yeah. But in principle, you would, you know the order of magnitude at least, but there is no precise time scale. Yeah.
The amplitude of the movement is what you expect.
Again, the amplitude of movement. You remember, it depends on there is a force. There is a constant 3 kT over gamma. So it depends on the force constant, it depends on the temperature. So at higher temperature, you expect larger. So the force constant is, again, a variable. So if what we have been doing is, for example, the force constant under the crystal structure conditions, if we can rescale it to have an absolute value in the crystal environment. So there are some functions, and Tim is going to talk about that, how to define the force constant. So you can get from there some absolute values. But again, I would interpret these things as within orders of magnitude. So I don't like to really make quantitative, absolute statements. Yeah.
But the scale of the biological scale is still applied. The motion of a residue will be much faster than that of an entire
Sure.
So it's still there.
Of course. So that is now the last thing I wanted to do, and it's going to be much faster, is the cross correlations. So if we go to, remember the cross correlations are expressed in terms of a heat map, right? So the cross correlation that we would like, if we want to show it, we already evaluated the cross correlation now. And we're going to show it in a figure, simply. The cross correlation based on mode 1 only. So this is the type of cross correlation that we obtain. So you can see from here how what does it mean? You have the cross correlation has been normalized here. So what it means is the delta Ri dot delta Rj has been divided by delta Ri squared times delta Rj squared square root of them, okay? Such that everything varies from -1 to +1. And this is the way the residue index has been defined. So you can see along the diagonal those red blocks. Why do we see red blocks? Because these regions are simply connected and they are correlated. They move together. But what is more interesting is we can see also other islands like that. So these are substructures that are not sequentially adjacent, but apparently move concertedly. Or the blue regions will be the regions that are anti-correlated, move in opposite direction. So it moves, it means this type of motion. Blue is like that, red is like that, obviously, okay? And finally, so we're going to write this to a file because we are going to switch to VMD again.
Is it typical to have that little amount of uncorrelated? I mean, it seems like there's very little green. Or is it just because it's a really small?
So if it's, how can it be uncorrelated? There are two different ways. When you take delta Ri dot delta Rj, there may be two reasons for that. One is if one is not moving at all, like it's located at a hinge site, no matter what the other is doing, it's going to be 0. Okay? And so these are the interfaces, like you know, between the passages from blue to red. The other possibility is, of course, if they are doing perpendicular motions. And we have incidences, but this is quite, by the way, this is based on mode 1 only. So if we were taking the superposition of all modes, things will disappear a lot. Yeah. Yes?
So let's say if you would take the 300 and change slices, I mean square for the correlation and stack them up, sort them up by normal mode, could you identify a conformational path?
That's a.
So if you had this in 3D, and if you could connect all the...
That we've been doing that a lot, actually, in many papers, and many other groups as well. So we try to obtain an allosteric communication path. Because now you know in this diagram that, let's say, you have a given, you know all the things that are correlated. Now you can start to go from one location to another to see what is being coupled. And then you have another group that is anti-correlated. And of course, the interface between them is very important for signal transmission. In the next lecture, and I don't know, I'm going really much slower than expected, I was going to show a case where the interfacial residues, if they are being perturbed, they completely obliterate the signal transduction. So it is, you know what are the particular sites that you would like to interfere with. The large majority of sites, single site mutations may not have any effect. But there are those critical sites that are important for signal transduction, for collective machinery, which we are able to identify by this type of analysis. Yeah, we've done this a lot, and many groups have been using this type of correlations to calculate, for example, in graph theory, you call these hit times, commute times between the different nodes. You would like to identify pairs of residues that communicate efficiently. And we use this type of expressions. So the thing is, you can do the same thing. You can load the file through VMD. VMD was already open here somewhere. So we have already written the file, and we're going to Okay, we need first to load. Actually, you need to load first. That's something. You know, the mistakes that I make are useful. It shows what you should not do. You should first select the A&M file itself before loading the heat map. HM stands for the heat map, you know. So let's do that again now. And then from here, you can load the heat map. So that allows you now to generate this heat map. Actually, it's in principle the same thing. But you can, again, you have the same type of correlations. An advantage is you can select from here any pair that you like, and you can view the pairs here. What are those particular pairs? So that is more or less what I wanted to do in the first lecture. And I will give a break now for 10 minutes, and we will continue after that. Okay?